Hey, well, welcome everyone. So thank you very much for coming. So today we have the pleasure of Ivana Bjedov from UCL giving the seminar. Ivana did her undergrad at the University of Zagreb, then her PhD in Paris, focusing on DNA repair and E. coli, and then got a very prestigious EMBO fellowship to go to Linda Partridge lab at UCL. He focused on the mechanisms of mTOR pathway and rapamycin. Drosophila, so she can see is moving up organisms from E. coli to Drosophila. And then she joined the UCL Cancer Institute as a group leader after obtaining an ERC Starting Grant, which is very prestigious, very difficult to get. And she's working on DNA repair and protein synthesis. And she's just become a CRUK Senior Research Fellow. That's a very hard thing to get. And it's also part of this elite environment. Today, she'll be telling us about delaying and measuring aging. Welcome. Look forward to talk.
Yes, thank you very much for a very divine invitation. I'm very excited to be here and share some fun facts. So my talk is going to be divided into two parts. In the first part, I'm talking about error in protein synthesis and how this can impact aging. And in the second part, I'll be talking about genetic clock, which we developed particularly for primary cells in culture to be able to investigate and detect and uncover some of the anti-aging drugs. So initially, I have just a few slides about aging, although I know some of you are very familiar with aging research. So this is to remind you how humans have always been very fascinating with aging and trying to find fountain of youth, as shown here in this old painting, where elderly would bathe and then they would come out rejuvenated. But scientists haven't attempted to tackle aging project until quite recently. And it's because aging is very complex. You have that gray, that wrinkles, every single organ in your body is deteriorating and every cell is deteriorating. So it was hard to imagine that you could find a single mutation that would revert the whole process. Until this paper in 1993, which was a work led by Cynthia Kenyon. So what Cynthia did, she took many worm mutants and she measured their lifespan. And she found that worms that had a mutation in insulin receptor, which is called DAP2, lived twice as long. So normally in the lab, median lifespan for worms is 20 days, whereas those DAP2 mutants have a double lifespan, 40 days. And what Cynthia Kenyon also uncovered in this paper, that this longevity of his DAP2 or insulin receptor mutants is dependent on DAP16. DAP16 is FOXO transcription factor. It's a downstream factor of insulin signaling. So if DAP16 is downregulated, then this exceptional longevity is abolished. But this was really the key paper that started this new biology of aging because it showed that a single point mutation, so this is a nucleotide change in the whole genome of C. elegans, that doubled the lifespan. So this showed that as aging is malleable, we can extend the lifespan by a single mutation. And another important thing is that what was established subsequently is that this is evolutionary concern. So initially, many have doubted and thought that this is a peculiar thing in worms, that this is would be quite difficult to translate to higher organisms. But now we know that a similar mutation in insulin signaling is incorporated into flies, for instance, cheek of flies. This is a mutation in insulin receptor substrate. They're also long-lived. And another nutrient-sensitive pathway, which is target of rapamycin signaling, when downregulated, also extends lifespan. And the key thing is that these are the major nutrient signaling pathways, and they need to be just mildly downregulated to achieve longevity and health improvements. Any severe downregulation would lead to embryonic mortality, and it shouldn't be healthy or long-lived. And the third key point that I wanted to make in this introduction is that anti-aging interventions improve health and prevent diseases. So this is illustrated on this image of insulin receptor substrate knockout. So these are the two sister mice. The one on the left is insulin receptor substrate knockout, and it's visibly up here. And I would like to highlight this is a broad spectrum prevention. It has the mice on the left has better glucose homeostasis, better motor function, better immunity profile, has reduced osteoporosis, cataract, and ulcerative dermatitis. So it's indeed a broad spectrum prevention of age-related pathologies. So this is from a review by Linda Partridge, when we contrasted this future signaling pathway in yeast, worms, viper, mammal, then illustrated how these age-related, the pathways that are implicated in aging are so well conserved. So yeast doesn't have insulin signaling as fast, but in the higher worms, flies, and mammals, both insulin signaling and mTOR signaling has been shown without regulated to extend lifespan. And this is some of this mutation mimic dietary restriction. We know that for more than 50 years, the dietary restriction leads to longevity, and it's been tested on many different organisms. So what Linda Partridge and Teresa Nicoli suggested is that traditionally we would take these age-related diseases, increase cancer, neurodegeneration, and develop different drugs separating proteins of those diseases. However, if those anti-aging intervention can prevent all of those pathologies that perhaps develop anti-aging drugs, we could prevent at the same time all age-related diseases. So they suggested the different strategies, therapy targeting the aging process itself, to simultaneously treat and prevent diseases. So initially, in this initial early years of new biology of aging, we've been mainly studying different mutants in worms and in flies and in yeast. The ones in the red are shown with that the becomes like a bit shock. So if you don't recognize the 6 kinase, or if you don't like the CHIK, or if you overexpress this C1 and 2, you will achieve this gentle downregulation of nutrients in body pathways and emotional. Or if you overexpress FOXO, this transcription factor, it will go to the nucleus and lead to longevity. And another point that I wanted to make about FOXO is that in some certain areas, it has been shown that they also have mutations in FOXO transcription factors. So even in humans, it looks like there is FOXO being important, although it's not quite clear. This mutation in FOXO that is present in areas, I think it's in the intron or regulatory region, so it's not quite clear how does this mutation manifest in those certain areas in individuals. So from this very genetic study research, the field is now moving more to the pharmacology. So that's why I'm highlighting, because now I'm in the Department of Pharmacology. So people are trying to find drugs that would target these very same pathways to replicate what has been shown before. So for instance, rapamycin is a drug that inhibits mTOR complex 1 and mTOR signaling. It's a gold standard. I'll talk about rapamycin later. It's a gold standard in aging research because whenever we test it on any organism, it has been shown that it's extends lifespan. Metformin is another interesting anti-aging drug that works by, it was predominant, the mechanism in LP, but most likely it works by activating AMP kinase. And the reason I'm mentioning metformin is because one of the first trials that might happen in humans, it's organized by Mir Barzilai, who would like to test metformin, give it to up to 3,000 elderly individuals who are aged between 65 to 79, and try to monitor them for many years and see whether regular taking of metformin and activating AMPK could reduce some of the anti-aging pathology. And the reason he picked metformin is because metformin has validated side effects. There has already been some clinical trials that showed when people take metformin, this would reduce incidence of type 2 diabetes. And there is some work in the UK that metformin was randomly given to some diabetic patients, and it was afterwards established that the ones who were randomly given metformin have much better regulated diabetes, have reduced incidence of cancer, so it was beneficial to have metformin as a treatment. So it has been used in clinic for a long time. So this is why Mir Barzilai thought it would be the safest to start with metformin in order to help. And obviously, the idea behind improving health in the elderly is because the life expectancy is steadily increasing about 3 months per year over the 160 years, which is a fantastic achievement for the medicine, and it's wonderful. However, the downside of this is that in every modern society, the number of elderly is increasing, as such, the number of people who are suffering from Alzheimer, Parkinson, cancer, and other age-related diseases. So here in the corner, I've put an image of Jeanne Calment. She was the longest living human. She died when she was 122 or 33. She stopped smoking, I think, when she was 111. But some people say she didn't smoke very much. She had very interesting life. For instance, when she was 19 years old, she had no inheritance because she outlived her daughter and she outlived her grandson. He died in a car accident. So she had a very small pension. She decided there was a system in France where you could sell your property, her big house, to someone, and then every month they pay a very small amount of your pension. This was about 400 euros monthly. Given that she was 90 at the time, to her neighbor, it seemed like a fantastic deal. She had a big house. But then 20 years later, the neighbor died. And then the widow had to continue paying this small pension to her. So it was the worst deal ever. So later there was some discussion in the literature whether she indeed lived for so long. So it can be about this, but most likely she did indeed die when she was 122. So my life is interested, the first time I have a talk, I would like to talk about translation actually. And the reason I was interested in translations in you is because in many aging screens that have been done, there is always this category about protein synthesis genes. That, for instance, this is a work by Matt Herbert and Brian Kennedy. Their lifespan is Pharmacology cerevisiae, and they measure lifespan in about 4,698 mutants. And the mutant that were long-lived, one of the group of mutants belonging to 6 years of ribosome translation and 4. So there is always this there is always this group of mutants that are implicated in translation that are long-lived. And there are other papers, for instance, by Planck and Papavi and by Cynthia Kenyon Marina Hansen. They downregulated in C. elegans some of the initiation factors, such as initiation E, ribosomal proteins, or S6 kinase, and this all made words live longer. But what was unclear to me when we started the project is what is the mechanism. So what was being suggested in the discussions of these papers is that perhaps if you lower protein synthesis, because this is such a costly process, perhaps you will save some energy which you can then reinvest to cell maintenance. Or perhaps if you lower the protein synthesis, then autophagy chaperones, protein degradation proteins will be more available to deal with any potential proteotoxicity. Or perhaps when you downregulate protein synthesis, you would translate some different genes, such as CAP-dependent versus CAP-independent. So the profile of the protein. But there was one hypothesis that was usually neglected, and this is that perhaps there are fewer errors in translation if translation is downregulated. So it's very easy to measure mutations in DNA, but it's very hard to measure errors in proteins. And we usually neglect errors in proteins because from a single messenger RNA, many proteins, and some of them will be quite transient, having a very short half-life. Some might have longer half-life, and they will be degraded. So the errors in translation are largely neglected. Also, it's very easy to sequence DNA mutation, and especially this is a review arguing that there is important role of DNA damage in the aging process. It's a beautiful spot there. Markers in the file. And also here, like illustrated, this is work from your colleagues from Cambridge, from Mike Stratt and Alexandrov. they completely redesigned the way we detect DNA and mutations. So when I was a student, we would just look that the C mutated to T. But what Alexandra decided, they were looking at the base before and the base after the C that is mutated to T, which gave us a much wider fingerprint and higher resolution where we could really understand which mutation might be causing these mutation processes. So when it comes to protein synthesis, the other end, so DNA polymerase would incorporate one erroneous nucleotide in 10 to the 8 that they're incorporating, whereas ribosome would incorporate one erroneous amino acid in 10 to the 4th that is translated, synthesized. So it's 10,000 times higher our ribosome errors compared to errors of DNA polymerase. Yet we are always neglecting the errors of the ribosome. So I thought it would be interesting to see and to investigate it and know a bit more about the implication of the errors in ribosome in the aging process. So we were just out into the first accuracy mutants that have been discovered in the 60s, starting at the beginning, and this was in E. coli. So most of the work on accuracy have been done in E. coli and mutants. And this is one of the most recent studies done by Vera Borbunova. So what they did, they took cells from different rodent species. So this is house mouse, this is some squirrels, and this is naked mole rat that I know the audience here is interested. So she took those cells into culture. She incorporated the reporter, dual luciferase reporter, which I will explain later in the talk, and it's a gold standard to measure error at the moment and protein synthesis. And she noticed that that mouse, which has a very short lifespan, has a lot of errors during translation process, whereas naked mole rat, that is exceptionally well made, only has much fewer errors during translation. Obviously, this is just a correlation, but I thought it was interesting. And there's some other work where in yeast, when these hypoaccurate and hyperaccurate mutations were introduced into mitochondria, and then correlated with being either short-lived or long-lived yeast. And this is work by Chris Sprout, who showed that that's some implication of the speed of elongation process in the accurate translation. So the faster translation elongation, the higher is the mistranslation rate. But what we wanted to do was not correlation study, just we wanted to directly manipulate the ribosome and the decoded cell. And we based it so we went to a lot of old literature, such as those by Gorini from the 60s, and they noticed that there was one particular ribosomal protein, RPS633, so ribosomal protein from the small subunits, 33, that is often mutated in E. coli in order to achieve either hyperaccuracy or hypoaccuracy, or alternative hyperaccuracy. And we think this is because this especially with this lysin 60 that is mutated. And I think this is because it's really at the heart of the decoded set. tRNA, messenger RNA, and when mutated by the arginine, asparagine, glutamine, threonine, this leads to hyperaccurate phenotype in bacteria and yeast. So at the time, I only had one PhD student working on the project, and CRISPR was a new technique. We wanted to do it in flies, which involves a lot of laborious, hundreds and hundreds single process to do. So we thought, how can we narrow it down from these 4 mutations? Could we find out which one would be the best to do in flies? So we thought maybe some of the long-lived organisms already have this mutation. So some organisms, such as bow-headed whale, with more than 200 years, and naked mole rat is also exceptionally long-lived. Hydra is thought to be immortal, and bats live very long. So we thought whether those long-lived organisms perhaps already have mutations RPS23. But to my disappointment, when we aligned the sequences, they all had a lysine at this position. And I would like to highlight how this is exceptionally conserved. So this is Drosophila, top is humus, and in this part of the RPS 23, there isn't a single mutation. So this is exceptionally conserved. This is really the heart of the decoding center that evolved over a billion of years, and there are no changes present. However, we found some archaea that have arginine in this position. So instead of lysine, they have arginine. And this is the ones in orange that have arginine. So this is only archaea, or this is zoomed in archaea. So only some archaea have arginine instead of lysine. And the archaea that have arginine also live in a little bit higher temperatures than the ones that have lysine. Although there are also the ones that have lysine that live in high temperature. So perhaps this very harsh environment of more than 100 degrees where these archaea live, maybe it's much harder to fold your proteins. And then on top of it, if you have some mutation, it becomes quite challenging. So maybe there is a selective pressure to be more accurate to facilitate protein folding. This is just a hypothesis based on some work by Brown and Wilker. We have no evidence for this. And also these archaea that have arginine, this mutation, they also predominantly do sulfur metabolism, they have acidophilic. We are trying to find how these archaea differ, the archaea with lysine. So we introduced this mutation, K6TR, to Pombi, to C. elegans, and to Drosophila, as in diabetes resilience. So hope you can appreciate here that those mutants that have arginine are much more resilient to the ones to the wild types, and same C. elegans and Drosophila. And then because we are at aging led, we did also lifespan, and we saw that also in place. So these are the one in orange and yellow. These are independent CRISPR lines, and they are longer lived compared to the wild type. So this is not a humongous lifespan extension, and that the 6 worms, but it's very reproducible, and we see it in 4 organisms that we tested. So then we wanted to see whether, like in coli, this is followed by regulation of errors input translation. So we use this dual citrate reporter, the same one that was used by Vera Porpunova. And these are the reporters that are the gold standard in the field. As I mentioned, it's very hard to measure errors translation, and this is the test that we left. So, and the control reporter, both granular and firefly, are expressed, where in the stop codon retro reporter, there is a stop codon in the linker region. So there is no firefly being expressed. However, if the ribosome does a mistake and the stop codon retro, then we will get some of the firefly expression. The more firefly you get, the more errors the ribosome is making. So what we did it in flies, in the young flies, we didn't see no difference between the wild type and the mutant. But in the aged flies, the errors went up in the wild type, but not in our mutants. So we think that this recapitulates something that has been seen previously in coli, that this mutation indeed is hyper-expressed. And we also lifespan those flies that have this reporter just to make sure that it didn't affect the longevity that are also not. We also measure translation. For this, we use pyromycin incorporation. So pyromycin is a molecule that resembles 3' end of amino-acylated tRNA. So it enters the A site of the ribosome, and it transfers to the growing chain and causes the formation of pyromycylated names actually release. So it basically, you get a lot of truncated proteins that are labeled with pyromycin, which is backed by the antibody. The reason we wanted to make a global protein synthesis is because some work by Maria Rodina showed that it could be, and others, that it could be that there is a trade-off between accuracy and translation. So the more accurate you are, the slower you get in translation. So in yeast, they saw that this is indeed the case. So this is incorporation of pyromycin, which correlates with protein synthesis. So our mutants synthesize less proteins. So it looks like that there is some sort of a trade-off. If you're more accurate, you synthesize your global protein synthesis is impaired. But this was more so in exponential phase than in the stationary phase, where POMB or discordant of POMB by the ACE is translated from there is a less demand for translation. So it looks like it's only during the high demand that our mutant is more complex. In C. elegans and Drosophila, these are the post-mitotic organisms, we didn't see any effect on translation. So it looks like in the metazoans, we managed to uncouple and we managed to improve the accuracy without having any effects on protein synthesis, so the global rates of protein synthesis. But to be sure, we also expected some of the larval tissue. This has been done in adults, whether this last work has been done in the larvae, and the larval, this this was done on wing marginal disc, which after metamorphosis gives rise to fin. So the reason we use this wing marginal disc, there are a lot of reagents available is in the actively dividing tissues. So like in POMB, where it looks like our mutant might be compromised more exponential phase than in the stationary, we thought if there is an effect of global protein synthesis, we are more likely to see it in the larval tissue where there is a lot of cell division and a lot of translation. So we use these tricks from Drosophila, where if here normally this heterozygous cell will give rise to two heterozygous cells, but if recombination, we use this mitotic recombination happens, if you use the case, then you will get two cells that are either that are homozygous. From heterozygous, you will get two cells that are either homozygous for your mutation or the wild. And this is marked here. So the overexpressed lipase just in the posterior part, the vaginal disc, using this H of Gal4 lipase driver. So in the anterior part, we had one copy of our mutant. And in the posterior, where we use this type of mitotic recombination, we had islet patches, clones of wild type tissue, clones that are JP negative of two copies of RPS23K6. And then when we did pyromycin experiment to see how much translate global potency, we didn't see any difference depending on how many copies of our mutants there are in the cells. So this showed that indeed in metazoans, our mutation didn't have any effect on the protein synthesis. We also checked some classic markers on translation, such as initiation factor 2A, phosphorylation in CD51, phosphorylation of S6 kinase, which is the output of TOR signaling. So the more phosphorylation you have, the more mTOR signaling there is, but this was unchanged. Phosphorylation of ADP was unchanged. So indeed, whatever we looked at translation, there was no changes except for the accuracy. We also measured the quantity in lives to estimate how healthy they are, and we have done on separate location of apparatus life, put it C. elegans, Drosophila, and when we went to compare our data, we both noticed there was a little bit of increase in fecundity. Here it's a live progeny of worms in worms, but this is egg-laying product. So it looks like this decline in fecundity that happens as the worms and fly age was a bit stopped, and they were laying more eggs compared to controls. Also, those flies had smaller wings. It was very subtle. It's known in Drosophila that all ribosomal mutants have a minute phenotype. They are very smaller, and those mutants also have a minute phenotype. But it's very subtle. It's very noticeable unless you do quantify them carefully. And they're also healthier. So we developed this Drosophila device. It was with gravity assays in the lab, so where you have to flip a single vial to a single vial. This allows us to flip 10 vials at a time. And we also use it here to measure health by negative geotaxis assays. So flies don't climb as well as they get old, so there is a decline in the climbing capacity in the elderly fly, but our mutant always perform better compared to control. So this means they are healthier. So now this is what I was puzzled, is because they were healthier, they were teacher resilient, they were long-lived, no effect on translation. So why don't we all have our gene instead of lysine? Because it seemed like all parameters look better in our mutant. And I think this is because this mutant is developmentally delayed. So this is data for Pompe. This is somewhat complicated graph for C. elegans, but this is the control and dwarfs that are 3.5 hours delayed and have similar phenotype like our mutant. So they're about 3.5 hours delayed. And then Drosophila is 24 hours delayed compared to normally it's 10 days for Drosophila from egg to the adulthood, the life cycle at 25 degrees. So I think perhaps this is why they are not more prevalent in nature, because they will always be outcompeted with the wildcats. So we also wanted to know whether some of the drugs that impact translators, such as rapamycin, whether they also have effect on accuracy. So we did this in S2 cells because it's much easier to work in cells than in microorganisms. So here we also had another report of formation operations, so ribosome can do different types of error. It could be stop-codon read-through, misincorporation, or a frame shift. And here we had another report of misincorporation. So again, firefly is here expressed but inactive unless there is a mutation that attaches. So the more firefly activity, the more erroneous the ribosome is. We tested the reporters using rapamycin. This is the drug that increases errors. And we saw a nice increase showing our reporters are working. And then we use rapamycin. So rapamycin is a drug that inhibits specifically mTOR complex 1. It doesn't inhibit mTOR complex 2. And as a result, we have down-regulated effect of acid kinase and up-regulated autophagy. And we saw that rapamycin did improve. There are fewer errors in cells that are treated with rapamycin. It's very subtle, but it's very reproducible. And as always in biology, it's much easier to make things worse. By treating rapamycin, it's easier to make a lot of errors, but it's much harder to make things better, as shown here by rapamycin. And we had similar results with taurine that downregulated the number of errors in the cells. And taurine is a dual inhibitor of both TOR complex 1 and complex 2. And we also tested trametinib, another anti-aging drug, which inhibits MEK-ERK pathway, and it's also shown that there are fewer errors. So we treated also mutant with rapamycin, and this extended their lifespan. So I think it's because rapamycin affects both autophagy translation, thus many more things. But there are mutants, just the manipulation of the decoding center that implicates, that reduces. So this is the conclusion for this first part, where we think that this lysine at the heart of the decoding center is optimal. It's involved for billions of years for evolutionary fitness. Whereas if we replace it by arginine, this is beneficial for lifespan and heat shock resistance, but it impairs development. And this is why this is not selected for evolution. This is good for health span, but not for evolutionary fitness. So we developed the first methods on hyperaccuracy mutant. We showed that one single point mutation can improve accuracy, and the control flies have a high stop-codon read-through when they're old, and this was reduced in the mutant. So we think this is, and we think this is developing. And then because this is pharmacology department, I thought at the end I will show a few slides about drugs that we did. So as mentioned, anti-aging field is now working a lot on different drugs. And in the Linda Partridge lab already in my postdoc, we showed that trametinib extend lifespan, rapamycin, and lithium. And when we combined all three, there was quite substantial extension. But it all revolves around inhibitors of the nutrient-sensing pathway. And we thought it would be nice to find some drugs that are unrelated, or at least to have a system that allows us to uncover new drugs that are not related to the pathway. So we thought that we could perhaps take advantage of this epigenetic clock that has been developed by Steve Horvath. So Steve Horvath in the 2013 published this paper that was so impactful. It has thousands of citations. So he found that we can use CpG site in our genome to predict the age of a person. So normally, we all have 28 million CpG sites now in every cell of our body. But if I take any tissue from you, and if I check only 353 CpG sites, I will be able to tell your age with probably 2 years error. So it's incredibly accurate. I developed a similar clock which was based on blood and 71 CpG sites. So it's quite extraordinary what is written in our genome and our epigenome and tell our age. So these epigenetic age estimators are a set of CpG sites that are coupled with mathematical algorithms to estimate the age in units of years. And it was a big breakthrough in the aging field. So how do this clock work? I think no one knows how they work, but they do work because now they're used, for instance, in forensic to determine the age of the cadavers, so they have a lot of issues. This is the small site, just a little bit to illustrate how these clocks are built. So normally methylation of CpG site, it's either methylated or non-methylated, but because we are looking at plural cells, so then there is different methylation levels. And some CpG sites, the methylation of some sites is EH can go down, and some methylations go up. So globally, we lose methylation a bit longer, but there is hypermethylation of some of the promoter regions. So some sites go up, some sites go down, and then there is a different rate. Some methylation of some CpG sites rarely change at all the age. Some simply decrease or increase. So then, to understand, to calculate the age of individuals, Steve Horvath developed taking into account what is the methylation value, and he combines this with the rate of this change of this particular CpG site as we age. And this is how the combination, the sum of everything will tell your epigenetic age. So this is where the epigenetic first clocks were developed. Now there are more clocks, from less chronological, more biological. But although Steve Horvath's clock is a chronological clock, if there were any deviation, according to his epigenetic clock, you are much younger, much older, it would indicate that perhaps you have some underlying disease. And for instance, this individual where the has accelerated aging, so the clock is ticking much faster. This is a healthy individual, which would the clock would tick slower. So there's this difference between chronological and now there are more chronological clocks. So what we wanted to do is, so Horvath's clock is amazing in telling your age, but it's not very good for cells in culture. So we wanted to develop a clock for primary human cells. We wanted to test them with different drugs and then see whether the clock ticks faster or slower. And then if we find a drug that slows down the ticking of the clock, to take it to Drosophila to see whether it works in vivo. It's because most drugs fail in clinical trials when that transfer to in vivo work because of the toxicity. So this is how all clocks, most of the clocks have been developed. So we took fibroblasts, primary human fibroblasts, dermal fibroblasts, and mammary fibroblasts. We passaged them carefully with no antibiotics for many passages. And then at time points, we took and examined the CpG methylation using Illumina EPIC profiles. And we pre-selected some about 2,500 CpGs where we used to build the clock model bioelastic, just as the Horvath did. And from this, we focus on 42 clocks that were selected to build our cell age clock. So here on the right, you can see that there are 19 sites that get hypermethylated through as the cells are being passaged, and some other 23 sites that are getting hypomethylated as the cells just, and some sites go up, some sites go down. And compared to the existing clock, multi-tissue clock, that is famous Horvath clock, so there was it didn't detect much age in the cells in culture, because this clock was trained to tell your age and the difference in years. So those clocks are amazing to what you train them. So forward clock is trained to tell your age, whether you are 1-year-old or 100-year-olds, but it's not very good for cells culture. There's skin blood clock, fetal age clock. So fetal age clock works in cells in culture, but we saw it's quite variable. Whereas the clock we developed is quite good in telling a different age in these primary fibroblasts that we trained them on. And then when we treated them with drugs, so this is predicted minus actual passage. So the control would sit here on the 0 axis. So this is minus negative because the predicted passage minus actual passage is negative. So they appear a bit younger. So at the 16, 18, 20 passages, the human dermal fibroblasts appear a bit younger genetically in their treated with rapamycin. And we also use human margaret fibroblasts. And we saw the same pattern with all of the drugs we tested. So rapamycin, trametinib, mTOR, this is known. And it's also known that taurine and those dual inhibitors that inhibit TOR complex 1 or complex 2 are less potent than rapamycin. And we see this in cells in culture that they were so good in reducing the age. And alpelisib, S235, this is a novel drug we uncovered in small but few experiments. This is a PHG kinase inhibitor. So we can see just a few months of passaging, we can see that the drug is anti-aging drug. We also look at different parameters, such as senescence, nucleolin, which is shown to be a marker for aging, IL-6. And we saw that these drugs also reduce some of the aging markers, classical aging markers. And when we feed them spliced, they extend lifespans, as mentioned. For some of it was known, but that policy will be. So we think that we can then take the primary cells, treat them with a drug for a certain time, inspect their epigenome, see how the CpG methylation has changed this time, and then using our epigenetic clocks, and this predict whether this drug slowed down the ticking or accelerated the ticking of the clock. And we hope that in future we could perhaps find novel drugs that are maybe a bit different, that affect proteostasis more than just pathways. And I have only one slide or two more, that no one knows how this looks. But because I'm in Cambridge and use Alpha, so they work a lot on reprogramming. So recently, much of the focus of epigenetic clocks has shifted towards examining them in the context of cellular reprogramming. So this is the conversion of somatic cell into pluripotent cell, the expression of Yamada factors. This can reverse the epigenetic aging signal. So this is done by Morgan Radin, who is now clearing usage. And when Steve Horvath inspected different hallmarks of aging to see which ones can revert and which ones can affect the clock. So when they used primary cells, which they immortalize by age 30, they still saw ticking of the clock. Genomic instability, so they irradiated the cells and the clock was still ticking the same. So, which was quite surprising to me that DNA damage didn't accelerate the clock, but I guess maybe it was, as I discussed earlier, it could be the cell type or the experimental condition. Nutrient sensing, of course, this down-regulated the clock. So this is the gold standard always. And they tested some drugs, some metformin cells, it didn't affect the genetic age, but this is all gonna affect that. So this is just to illustrate how, despite it being fantastic invention, those epigenetic clocks, there is still a lot to be understood about how they work, and hopefully this will help us understand the biology underlying the aging process. So that's all from my lab, and thank you for listening. So this is the first part of the work was done by Victoria, fantastic PhD student who did all this ribosomal stuff like Xenia, Krista, and Ellie. And then this second part of the project about the epigenetic clock, we met some brilliant collaborators. Stefan Beck, who knows everything about epigenetics, who helped us with primary cells, and Prof who developed the epigenetic clock. He's an amazing bioinformatician. So thank you for listening, and if you have any questions, please ask.
Well, very exciting. Any questions? There's been a lot of research into peptide calorie restriction.
Yes.
Given that I was thinking insulin, do you think that acts very similar mechanism to what you've been?
Dietary restriction, there is rapamycin considered to be dietary restriction method. So it was being proposed that perhaps instead of calorically restricting yourself and suffering, that maybe you can just take rapamycin, which would have some overlap with dietary restriction. And I think recently there was a big study of dietary restriction, which shows surprisingly there is a lot of impact on the genome, not just the metabolic, because it was always considered the dietary restriction or caloric restriction while impacting metabolism and reducing having better profile. But looks like the genome is also very important. It was just 10 weeks ago published in this large study on mice. So it was very interesting. Yes.
Adam,
yeah, thank you very much for your talk. Following on from that, you mentioned about the big study on mice. Have any of those looked in the context of cancer? Because you know cancer is generally a disease of age. It sort of delays the onset or delays the progression of cancer.
Yes, there was some study. Now I've blocked for the name, but yes, there are some study in mice showing that some of the anti-aging interventions would delay occurrence of cancer. There was a lot of this being done in caloric restriction. And also in worms, I think the first slide I showed about C. elegans, these stuff to worms, I think where they were crossed with some ovarian model of tumors in worms, then they didn't develop the tumor. I think there were more apoptosis in those ovarian models. Actually, it's a germline model of tumor in worms.
About how the genetic clock works in cancer cells in general, which are, like, strictly speaking, immortal. How does it change it?
Yeah, that's very interesting. So if you read the first paper by Horvath, so remember, as you read, it's a brilliant paper. He's the sole author of this paper. And as you read it, you start thinking, oh, what happens with iPS? And then he did it. Oh, what happens in cancer? So it's a really nice paper. So I think he showed something about cancer cells, but I think with cancer cells, because there are different mutation, different tumor, depends where you look. I think originally it depended in his paper of status of 353. So when there were 353 patient, but I remember it was erratic on this. I think with cancer cells, you can find tumors where the epigenetic clock is accelerated and decelerated because it's almost up. But so I don't think there is a consensus. But in the original paper that was published in 2015, he reported that this depends whether the clock will tick faster. It will depend on status in the... Yeah.
Sorry, it's another cancer-adjacent question. But I was wondering if, when you're looking at mutations in the RPS, whether you saw any mutations that were correlated with hypobacterial translation, and whether that might feed into context like cancer.
So we wanted to do hypoaccuracy. We did now in yeast hypoaccuracy. So the hyperaccurate, they're always in this RPS 23, usually in the part of the decoding center. The ones that make more mutants, they are S4, S5, they're different domain of the ribosome more often. But also in RPS 23, there are also some ribosomopathies in humans that are reported when there are adjacent amino acids that are mutated ribosomopathies. But yeah, we wanted to do exactly this, to go the other way around, to increase the mutation to see whether the lifespan will be shorter. But it's really hard because of this effect on development. We tried to make another very potent mutant that would reduce errors, but we never managed to get those CRISPR lines in place. So it could be that we are just rubbish, or it could be that I think it's because it just affects development too much. So maybe if we bypass development, just do it in adults, maybe we will be able to do it. So
I wonder how the proteostasis is going with your improved accuracies. I think that will obviously lead to a better proteostasis, less of the aggregation of unbound protein and stuff like that.
yeah, this is all things we would like to monitor. So in the papers, they did some of it in CLNS because they have good. So we joined with from organism because just CLNS has a good reporters. They have this heat shock GLP reporter. So they use heat shock 16 GLP reporter. And I think when we heat shocked, we heat shocked them and we treated them with pharmacomycin. So I think when we treated with pharmacomycin, the wild type of worms, a germ mutant, there was less of an induction of this reporter, which interpreted that they're probably more resilient to proteostasis because pharmacomycin would induce a lot of errors in translation. But yeah, so we did a little bit of this in work, but during the revision process, it was very superficial. So we would like to do more. I think this is a spot on question. Yeah, this is what we.
Asking question. So are there any, in the first part, so are there any genes or proteins that are known to be more prone towards liposomal errors?
More. Well, that's. I would assume if slowing down elongation step of translation leads to fewer errors, I would assume that perhaps some oncogenes that accelerate translation might lead to more errors. And I know that cancers have upregulated HSP90 very often, so maybe for other reasons. But I would imagine that if we push the translation to be a bit faster and that some oncogenes might do the job.
And has anyone ever looked to see if the errors are a factor of tRNA abundance or
Yes. I don't know. Well, there's also the factor of usage, of the codon usage. So Judith Friedman, I think she had some beautiful review where she was saying that if you have a let's say catalytic site of a protein, usually you have some tRNAs and the codon which have a heavy usage to make sure that tRNAs that are required to translate this catalytic site are very abundant. So there are no mistakes because if the codon tRNAs are very scarce, you might incorporate the wrong amino acids. But on the other part of the protein, which might be quite challenging to fold, you might have some rare codons, kind of to slow down a bit translation to allow the folding to happen either in the ribosome exita or after, so that and people have seen that if you want to overexpress your protein in E. coli, if you just have optimal codon usage for everything, that sometimes here and there you need some rare codon just to slow things down to make sure it's sold properly. So the codon usage is quite interesting in the tRNA abundance, but I think there are more people who know more about tRNA abundance than me.
Wait, any more questions? One final question.
Probably just a more general one. Like any of the medicines you studied, can you just make any best continuous for daily supplements or that in age?
Any of the drugs I mentioned?
Like, for example, like I've seen a lady had as supplements for people as an anti-aging drug.
Yes, there is Lenny Guarente in MIT who have the supplement. Elysium is a company, and I think they sell some supplements with antioxidants and NR or something. But I have no idea what the effects of this are. I read one interview about some people taking it and asked someone else, the journalist asked them what are the effects, do you feel better? And one said, oh, the skin on my elbow is softer. And the other person said that the nails are growing a bit faster. So I think this is very poorly researched. I wouldn't recommend these compounds.
Great. Well, don't be taking that one.
Yeah.